We are in the process of building a catalog of genetic abnormalities for cancer. And in the next 5 to 10 years, I think it will be possible to sequence large portions of the tumor genome for $1,000. $1,000. We will be able to predict hotspots in the genome with SNPs, and we're able to do this very quickly. It'll be done, it'll be automated. And it'll be done in places like Columbus, Georgia, where we're send a laboratory a small snippet of tumor in a fashion that that tumor will be preserved, and it'll go to the lab, and that data will be sequenced right then, so we have it for use. In addition to looking at the genetic profile, we'll also be able to look at the protein expression coming from those tumor cells. And we'll be able to actually look at blood, and we're now able to capture in a blood sample 2 or 3 or 4 circulating cancer cells within a specimen of blood. So for patients who have breast cancer and colon cancer, and you want to predict how well they're going to do, there are commercial tests now that can detect as few as 1 to 2 cells per milliliter in a blood draw. The higher the number of cells, the greater the likelihood for metastatic disease. The lower the number, the better people do. This is the future of what your pathology report is going to look like if you get a diagnosis of cancer in the next 5 to 10 years. It will no longer be a written description by the pathologist, but it's going to look like this. It's going to show aneuploidy, some gross genetic abnormalities. It's going to show copy numbers, genes that are amplified. It's going to show single point mutations and hotspots in those genes. It's going to show genetic splicing alterations, how genes come together differently. It's going to show how the DNA is modified by methylation. And it will show a pattern of genetic activation or downregulation. So this is the future of what our pathology reports are going to be looking like in the next 5 to 10 years. Once you have that, you move away from describing a tumor by looking at it into describing a tumor by the genetic morphology and how it actually grows. And you can actually create a repair manual for the defects in that tumor. You understand passenger defects, meaning things that happen epigenetically, versus true defects that are driving tumor growth. You'll be able to determine which genes are critically important to drive growth, so-called oncogene addiction, and find genes that are amplified but not really relevant, and you won't target those genes. And this is the paradigm that Dr. Niederhuber has. Dr. Niederhuber is the head of the NIH, for how all of this is going to come together from academia and the universities, in the community cancer centers, and the cancer centers, and out further into the community. We will have a data storage center where we will collect patient data and tumor data, which we're already doing now here. This will go to the NIH. It'll come from places like the NCCC, of which we are a part. It'll go to SPORES, which are sites that, cancer sites that focus on a unique disease. It'll go to CCOPS, of which we're a member. And then that information will come back and forth to the physician, whether he or she is practicing at a university center or in the community through one of these organizations. In summary then, to finish this part of my talk, whole genome sequencing will be a part of cancer care in the next 5 to 10 years. And we think that by the next 10 years, every patient that gets a diagnosis of cancer will have that ability to have their tumor sequenced and that data stored down the road, should it's needed, to modify therapy. Personal genomics will lead to a transformation of medicine. And it's not just going to happen in oncology, it's going to happen in hypertension, it's going to happen in diabetes, it's going to happen in a wide variety of diseases. We will have web-like connectivity of the kind that I showed you, where a center like ours will be connected to the NIH and vice versa. And we will have collaboration. We'll have to have collaboration between the government and private sector to mediate these kinds of affiliations.